The overall goal of this procedure is to quantitatively determine the transport activity of reconstituted chloride channels or transporters. This is accomplished by first reconstituting the purified transporter into liposomes at a low density, so that each liposome contains either 0 or 1 copy of the active complex. After removing the external chloride ions so that there is an outwardly directed ionic gradient, the liposomes are added to the recording chamber where the chloride concentration is monitored using a chloride-sensitive electrode. Next, chloride efflux from the liposomes is initiated by addition of valinomycin, a potassium ionophore which relieves the membrane potential. Once the signal reaches steady state, the experiment is terminated by the addition of detergent to dissolve the liposomes. Ultimately, by measuring the rate of chloride efflux from proteoliposomes reconstituted with a purified channel or transporter, it is possible to quantitatively determine the unitary transport rate and the stoichiometry of the active protein complex.
The main advantage of this technique over other existing approaches, such as fluorescence-based flux measurements, is that with this technique, it is possible to quantitatively determine key parameters of the reconstituted transporter, such as the unitary turnover rate and the stoichiometry of the complex.
In addition to providing quantitative measures of these key parameters, this method can be used to rapidly and precisely determine the effect of mutation, small molecules, and modulatory subunits in the reconstituted transport protein.
The recording setup consists of 2 chambers, a chloride electrode, a pH meter with an analog ore.